Okay, here we go for the last session of this week, session 5 on sustainable, I hope, ocean innovation. We're going to have Mathilda, Valerie, Lena, and Jakob. We're really eager to go today, I'm sure. I would like, as usual, to thank your reviewers, so the different members of this jury who reviewed your papers. Dominique Adriens from Ghent University, Steve Como from Sorbonne, Love, Robin Fayetaz from Ifremer Lorient, Nadir Lafayette from Université Côte d'Azur ICN, Keila Plichon from Université Côte d'Azur IRCAN, Juliette Vilchanou from Coral Guardian, and Bertrand Wenling from Satouin. And so for this jury today, on site and online, Robin Fayetaz from Ifremer, Aziz Moula, and maybe for the last time, for the last time, I will say it this week, sorry, from Rudyard Boschkovich Institute, and Juliette Vilchanou also from Coral Guardian. We are all here, ready for you. So I guess you are ready as well.
I'm ready.
All good? So the floor is yours.
Thank you. So hello, everyone. I'm excited to present my master thesis today, Cultivation of Baltic Sea Macroalgae, where I focused on growth performance and hatchery optimization of Ulva species. So I did this project with my wonderful supervisors, Josephine Larsson and Gonzalo Carneiro, at the Marine Center and Soto Ocean Culture in Simrishamn, Sweden. So seaweed has been produced and consumed in Eastern Asia for thousands of years, but today this industry and the interest for it is growing elsewhere. In Sweden, seaweed cultivation is really at its infancy, and there are only a handful of companies here operating on the west coast. But what about the Baltic Sea? Despite this very long coastline to the east, there's no cultivation of seaweed here at all. This is mainly due to the fact that the Baltic Sea is the world's largest brackish water body, and it's because of all these 200 rivers that discharge into the Baltic Sea, which leaves this pretty impressive gradient of salinity from 3 PSU, so the practical salinity unit, and up to 20 PSU around on the west coast. What this means is that there is a much lower species diversity here compared to the west coast, and we can't simply cultivate the same species here. Despite these challenges, let's say, Soto Ocean Culture is a startup that is piloting seaweed cultivation in the Baltic Sea. And this is, again, the company I did my project with. So the idea is to have a land-based cultivation system where you have a flow-through of natural seawater from the Hane Bay, which is situated here in the south. But like I said, you can't just cultivate any species in this part of the world. So this is where Ulva prolifera comes in. Ulva is a green algae that you can find basically anywhere in the world. It grows fast, and in Sweden, at least, it grows in the summer months and usually in rock pools like these. The setup for this land-based production looks like this in a simplified way. We have a hatchery phase where we induce reproduction in Ulva in order to create these beautiful little clusters that we can later put out in large tanks outside. So this is where we get our biomass and they grow big and nice. This you can harvest and eat, and a small part of it will also go back to the hatchery where we can induce reproduction again, and this is how we close the cycle. But this hatchery phase is very important to control for any seaweed producer. And most of the time, it's environmental parameters that affect the timing and the efficiency of the reproduction of Ulva. But since Ulva are so plastic and a very adaptable species, these specific triggers for reproduction vary a lot depending on where you are in the world, but also depending on tides or the strain level even. So since there's no cultivation of this in the Baltic Sea, there are no protocols that are established for this reason. And that leads me to the aim of my project, which was to help developing pioneering Ulva cultivation together with a startup, Soto Ocean Culture, in a small scale and commercial setting. So what we wanted to see, and our research question was to see how do different temperatures influence zooid, so gamete or spore release in a local strain of Ulva prolifera. And in order to detect that and to compare, we also wanted to develop and validate an accessible and reliable method to quantify zooid release in these species. I will just add also that I looked at the growth rates and the temperature effects on those two, but for the sake of time management, I won't talk about that today. But I'm happy to discuss it after. So the presentation structure will look like this. I did a first trial with a laboratory acclimated culture, and we faced a lot of challenges, let's say, which helped us optimize and try again with a wild culture. So I will present both of them and in this order. But first, we need to know a little bit about ulva reproduction. So ulva does not have any reproductive organs, unlike like some other species. Instead, these green and vegetative cells can completely transform into reproductive cells by changing color and creating these zooids. And these zooids are either zoospores or gametes. And I will talk a lot about them, so keep that in mind today. And when they are ready, they get released from the tissue, which leaves the tissue white and essentially dead. So the cells are empty. And this is very important to understand, because if this happens spontaneously in your cultivation or in your production, that can lead to a lot of biomass loss. But in the hatchery, of course, we want to induce this reproduction because we want them to reproduce. And it looks like this. So to induce zooid release, you cut the apical part of an ulva thallus into small fragments that you rinse with some distilled water. And essentially, this stresses the fragments, which leads to them becoming reproductively mature. And again, they change the color to this brown or yellowish color. After some more time, the zooids are ready to be released, leaving the tissue white and empty. And these zooids are phototactic, so they swim towards light, and that way you can collect them and use them in the next step of your production. So by doing that, let's get into the first trial with the laboratory acclimated culture. So we did the steps that I just explained, and with a room temperature acclimated stock culture of this Ulva prolifera. This was divided into 3 groups with 5 replicates in each treatment and around 30 fragments per dish. And they were incubated in 3 different temperatures. So we had a colder temperature, a room temperature, and a warmer temperature. And to quantify the zooid release, we initially aimed to develop an image analysis. So the idea here was to take photos of a subset of fragments in each dish daily to track the color changes when they go from green to white. And the analysis was done in image J, and simply the idea was to quantify the amount of white pixels compared to darker pixels. So let's see how that turned out. First of all, we could observe this zooid release in all of the treatments on day 4, so simultaneously. However, we had a very large variation within each treatment. So this is an example from the hot treatment on day 4. We had some fragments that were, they remained green and vegetative, some other that had engaged in these outgrowths, and some had started to release their zooids or almost completely discharged the zooids. Additionally, we learned the hard way that we had some issues with diatom contamination. So this is how it could often look like after some days of the experiment. We start with this green and nice fragment, and after some time, these microalgae has started to grow, and they get a brown coverage that is not due to maturation, but rather diatoms. That, together with extensive outgrowths in some of the fragments, ultimately led to the fragments becoming darker instead of white, which was a bit troubling. So even in cases where we saw and we could observe zooid release occurring, this is what you can do here. So all the little dots are zooids. The fragments remained and had this dark color because of these these effects. And if we compare to what we had expected to see, it was quite clear that the image analysis wasn't strong enough or developed enough for our case. And it was also importantly partially due to the fact that the stock culture was in a very poor state. So all of this led us to think that we need to try something else and see if that will work. So that leads us to trial 2. This time, we harvested fresh ulva from the sea, and they start coming up around April in this area. So at the time, the sea was very cold. It was around 6 degrees. So we decided to acclimate them in 15 degrees in our fridge instead of in room temperature to remove a huge temperature shock. But other than that, the experimental setup was the same. We had our 3 temperature groups with the same amount of replicates. However, this time, we only had 10 fragments per dish instead of 30. And we also did a daily water change in all of the dishes. And this was to minimize diatom growth. And so to quantify zoid release this time, we tried another approach, which we call the visual scoring method. And here, we simply looked through a stereo microscope at the tissue and detected the color changes by giving them a score. So here we looked at both the green to brown color change, but also the brown to white color change. And since we had 10 fragments only in each dish, we analyzed all of them daily. And the scoring looked like this. If 50% or more of a fragment had become brown, it was considered mature. And if 50% or more of a fragment had become white, it was considered discharged. So let's see how that turned out. This time, we could see very distinct color changes in the fragments. And this enabled a more successful analysis. So the first picture here, you can see a green and a vegetative fragment next to one that is reproductively mature, that has changed the color. And the other 2 fragments represent those who would have been classified as discharged because 50% or more had released their zoids. We also, importantly, had much less problems with diatoms this time. And it also enabled us to do some statistical testing. So here we have a graph showing the days on the x-axis and the percentage of fragments on the y-axis. So it's the percentage of mature, so brown fragments with the dashed lines, and white and discharged fragments with the solid lines. And we have our temperature treatments. So we can see that the cold treatments started by day 3, already 36% of the fragments had become mature. And in the end of the experiment, almost all of them had discharged their zoids. The room temperature had a bit slower response. So here, 18% of the fragments had become white on day 4. And in the end, only around 50% had released all of their zoids. Interestingly, in the hot treatments, nothing happened. So they remained green throughout the whole experiment. So the fact that we have quite contrasting results is interesting, because in the second trial that we just observed the graph, we saw that we have significant and very different responses to the different temperatures. Whereas in the first trial, even though we couldn't quantify it because we had issues with diatoms, etc., we could still observe that it was occurring on day 4 in all of the treatments. And what this could suggest is that maybe there's not a magic or absolute threshold that triggers reproduction in these strains, but instead a relative temperature shift that could trigger it. And this is also supported by literature, and some researchers have found that for other Ulva species, an increase in 5 degrees could be a good rule of thumb to induce reproduction. But it's very important to highlight as well that we use different protocols because of the optimization of the protocols, but more importantly, we use different starting materials, so we cannot directly compare these results. For the method development part, the visual scoring method proved way more reliable and easy to use, frankly. And this was simply because you could observe these tissue color changes, even if we had some diatoms or if the lighting was a bit off. You could still observe the color changes in the tissue compared to the image analysis. And also, this noise that the diatoms caused in the image analysis is something that is quite frequently discussed in similar studies as a big challenge. But of course, there are some limitations to the visual scoring method as well, and it's the scale and subjectivity. So, of course, we had now our 50% threshold of yes or no if they were discharged or mature, but adding a narrower scale could potentially increase the resolution of the results, but that would also invite more subjectivity and potential human error. So the take-home messages from this experiment to conclude would be that zoid release is potentially driven by a relative temperature shift rather than an absolute threshold. However, this needs to be further studied, of course. But what this could mean for the production directly is that adding a gradual acclimatization step in the protocols in the production will be important to trigger or to control these triggers that are caused by the environment. And that will both help to optimize the production in the hatchery, so you can control the zooid release, but also help prevent the spontaneous zooid release that can lead to biomass loss. So some next steps for this project will be to actually apply all of these results to the pilot production that is ongoing right now. And I think a next step will also be, it will be very interesting to check how light and nutrients affect the hatchery phase and, of course, the growth phase as well, specifically because these fluctuate a lot and are quite specific to well, Sweden and the north, and Hane Bay specifically. And finally, developing targeted diatom contamination mitigation protocols will be key to have this a successful cultivation. And with those steps, we're ready to continue working on this. So thank you very much for listening.
Thank you, Mathilda, and thank you to Joséphine Larsson, so Gonzalo Carneo, your supervisors at the Marine Centrum
And Soto.
And Sweden. Sorry, it's better if you say it. Thank you. So yeah, with this session, we are going really in the practical aspect, in the production, how to make it operational. And for the questions, we are talking about algae. Let's start with Aziz. We're on time, so Aziz, and then we'll go to Juliette.
Thank you very much. Super interesting. And to make Ulva interesting is a pretty good feat as well. So well done. I was reading the abstract, and one of the sentences, and you mentioned it in your introduction, is, you know, in Asia this has been happening for thousands of years, but then you go on to say we have a lack of established cultivation protocols.
Yes. No, it's true. And I wanted to highlight the fact that we often talk about seaweed as something so new and innovative, but it's actually something that we have a lot of knowledge about, and it has been done in other parts of the world for forever almost. But it's very specific to the Baltic Sea. And for this specific region. And since specifically Ulva is also so adaptable, so there are so many papers about Ulva, but the temperature, the preferred temperature always differs. Because it's so different depending on species and even strains or where their natural habitat is.
Okay, perfect. So I'm super interested in this, and I really like the result of the shift in temperature rather than the temperature itself. So where was the shift? Was it from when you collected it to when you put it into the lab, or was it from in the lab? Did you keep it at a constant temperature? Sorry if I've missed that.
No, but it's a very good point, because when we had the lab acclimated culture, it had been in 20 degrees roughly. And then the shifts were not that big, to 15, staying at 20, and then up to 23, 24. But the one that we collected from the sea, of course, there's going to be a huge shift from 6 degrees and coming up to even 15 degrees, which was our acclimatization temperature. And this, I think, definitely affected the results. So it won't be like from 15 and then...
For sure. And I'm trying to not think in a kind of ecology way, but more in a kind of practical way, that this is a good thing, that you know that this shift in temperature, you can control the release, for sure. But I mean, for the 28 degree one, there was 0, right?
Exactly.
Do you think the shift was just too much?
I do. And I have a theory. Let's see.
Because I mean, going from 6 degrees to 28 degrees is...
It's a huge shift.
It's a huge,
of course. And it's often talked about this, that usually there's a span, an optimal span. And within this span of temperature, you can induce zooid release, for example. And it's the same for growth. But if you go below or above, it's too much for the species. So this one that had the biggest shift, it remained green. But we could also see that it had accumulated this starch granules, which indicates that it changed its metabolic pathway.
Because that's not climate change, that's like world ending. It's good that you tested it.
No, for sure. But it's also important to think that where these species live, it's frozen for some time of the year. And then the water is low, so they're dry for some time. and then they grow in the summer when it can go up to 22 degrees, perhaps 23,
Okay.
maybe even higher.
So it tells us that their temperature range is huge, not their shift range. Yeah.
Exactly.
So you kind of induced some synchrony because the number of fragments that released at the same time was the same. They are released. Yeah, they're released.
Exactly.
So in your protocol that you'll develop, then you have synchrony, you have the protocol already that 100% will release.
Yes and no. So this was really, really nice to see that they were all acting pretty much the same. It was very much like they knew what I wanted them to do, and then it all just happened. But I did this many times before, and it did not work like that. So I think one reason is that the fact that when you pick it from the sea, it's very easy to see where the end, where the apical part is. And this is the part of the ulva that is most reproductive, and that's where they release their zooids. But in the cultures that you keep in the lab, when they grow, it's they're free floating, and it's a little bit harder to see and detect this apical part. And that's why I think we had, one of the reasons why we had such a large variation in the first experiments, because it wasn't as easy to find the actual right part of the ulva, let's say. So I think that also has an effect on the results.
Okay. I have a couple more questions. And you mentioned it, so I'll ask it. The phototactic stuff. They're not phototactic throughout the development stage, right?
No. So actually, we have.
And are they all phototactic?
No, actually
some
not.
Will be opposite, right? I'm guessing.
Yes, and this is where it gets a little tricky, because ulva has different types of reproductive strategies. And so some use sexual reproduction, and then they have a diploid sporophyte state, and then a haploid gametophyte stage. And so the sporophyte is the one that is releasing the phototactic zooids. But the gametophytes, they release gametes, and these are actually, they go away from light until they find each other, and then they're phototactic.
Yes.
Yeah.
So the reason I ask is it's like a whole study in itself. The reason I ask is because this will really affect your protocol a lot by the number of individuals that you're able to culture as well. So it's important. I mean, actually both end in the same way. You can still collect them just at different ends of the test tube.
Exactly. And that's where, when they release the zooids, when you notice this, you put a light just on the side of the petri dish. And if this happens in a big quantity, like all of them do this at the same time, you can actually see them having this little green, yellowish color on the side. And that's the one you will pipette up and use for the production.
Just a quick question. Can you not do this in situ? Can you not just do it in the rock pool?
In the sea? Yeah, the idea is to have the land-based production. Yeah,
of course, but I mean, you could do this in the.
You mean the reproduction?
The reproduction.
Yeah, although when these zooids are released and when you pick them up, you kind of isolate that specific strain. So in the sea, and that is something that we noticed, for example, here you see that there are some things going on in the bottom of this dish, but it's other microalgae, other things that grow. So that's the problem.
Sink?
The other microalgae?
Yeah, the zooids, do they sink?
The zooids? No, so they swim towards lights, and then you pick them up. And the idea is to put them in, well, they always will go towards lights. So that's, you put them in darkness after you've collected them for them to sink and then attach to the bottom of the dish.
So what is the natural timing of their reproduction?
They reproduce several times, if I'm not wrong.
During the time of day?
I think at night.
At night, okay.
Yeah.
Okay. All right.
But I'm actually not 100% sure when they do it naturally.
And just, I'm sure other people will ask, just can you say something about the temperature and this timing of growth? What was your kind of main result in the timing of growth that you couldn't?
The growth?
About?
Yeah. Well, let's see. It looked a bit like this. So actually, to just don't mind the text. To summarize it, different temperatures didn't have an effect on the overall accumulation of biomass. But when we looked at the relative growth rates, the warmer temperature peaked quickly, but then they decline, whereas the cold temperature kept more of a steady growth curve. And this is good because temperatures will fluctuate a lot in this area. There's a lot of upwelling, and so you can go from having 20 degrees one day and then all of a sudden it's 9 degrees in the water. And since we're using the natural water from the sea for the growth, we were quite happy to see that it didn't have, this time at least, a huge effect on the overall growth.
Thank you.
Yeah.
Thank you, Aziz. So we can go to you, Juliette, and we'll finish with Robin.
Hi, thank you for the presentation. It was very clear to understand. I have a bit of question on more the company aspect to understand a bit how your findings might help. So you say in the first experiment that it's more like lab and hatchery based. So everything changed, like so it got released on day 4, but you saw lots of changes every time for each temperature. And all the problems you had, it was always the same for each temperature, or was one still a bit like less worse than the other?
Yeah, no, I understand. So they were all quite contaminated. So all of them had a lot of diatoms and other things. It was a bit messy, but the diatom growth was slower or not as significant in the colder temperatures. So that's also something I discuss in my thesis, is that keeping colder temperatures in the hatchery phase might also mitigate diatom contamination or diatom growth. I think it's very hard to remove them completely because they live together.
And how long was this batch of hatchery that you use already there, like in the big tanks, I guess? So you took it from the big tanks. How long were they already being used?
They had been picked in 2024 from the sea, and then they were isolated this way with the Zoid release. And then they had been growing in tanks the last summer, let's say, and then picked up and put in the lab. So they had been in the lab for maybe, let's say, 8 months or so. But yes, so they had been there for quite a bit.
And what do you think could be a way to try to minimize all these problems? So you said in the discussion that would be like another study to try to see how to do it, but do you have ideas already, like renew the tanks every year, or I don't know?
Yes, no, for sure. That's something we discussed a lot, that actually keeping the stock cultures healthy turned out to be such an important, like, it sounds so simple, but it turned out to be such an important thing that we learned. And I think just changing the water more regularly, giving them some nutrients and more light, and perhaps also keeping them in colder temperatures, because they've been kept in room temperature, and the lab, well, the room that they are in, the temperature there fluctuate quite a bit because of, well, the seasons and the sun and everything. So maybe keeping them in a more stable and colder environment, I think, could be a good idea.
To be able to get the results you got with the wild ones, but in the...
Potentially, yes.
Yeah. Okay. Yeah, then I had questions, but Aziz asked them, so we can move on.
Let's go to you, Robin.
Thank you very much for the presentation. It was very interesting. I mean, I really don't know much about all this, but it was very clear and very interesting. Well, Aziz asked most of the question, like temperature shift, and so I will go back to the image analysis. So basically, your conclusion is that you need to discard it because it was less. I mean, it was less efficient, at least in your case. Do you think it's to fully discard because your I don't know, your visual score was much better, or do you have any ways to improve it?
Yeah, no, I don't think that it had to be or has to be completely discarded, but we had some additional problems apart from the diatoms with the image analysis, and it was, for example, that well, Ulva, I didn't.
Now?
Yeah. Oh, you don't see the screen? Okay.
I'm sorry.
No, I don't know what. Yeah. Okay.
Oh yes. No, that's Ulva prolifera, which we used, is tubular, and so it has gas inside, or oxygen that it produces. And so, for example, these bubbles. Oh, now you have another. Let's see, maybe like this. Yeah, like bubbles inside of the tubes affected also this, especially when you look at the darkness of the pixel, and that is the part of the analysis. And also the fact that they were tubular led to them having these overlapping areas where they would be darker naturally, even if it wasn't because of zoid release, but it was rather because of the lighting. So I think either cutting them differently or having another way of preparing and standardizing the samples could be a good way of also trying again. But yeah, I think, and also perhaps not looking just at the pixel intensity, but also the color change. So having like a color scale of RGB, right? The red, green, blue scale. And using that instead of having the black and white protocol could maybe also be an idea to continue working on it.
And do you think there would be other approaches of image analysis that could be potentially less biased or less sensitive to the parameters that you set in image J?
Yes, I know there is one study that has been done in Sweden as well, where they looked at Ulva fenestrata. So already it's not a tubular version of Ulva. It's another species that is like a flat. And they used image analysis to detect, well, they looked at the green color nuance and correlated that with nitrogen content. And that could be used to detect how much protein you have, for example. So it was also kind of an applied approach using image analysis. So I think that could be really cool, and I would like to test that with this Ulva.
Thank you.
Good. Merci, Robin. So we can open the floor to your questions to see if you're still so active today.
Sophia?
Thank you, Gwen. I wanted to ask you if they already had a plan on how to sell these algae, for what they would be used. Like you mentioned that it's probably for to eat them, but like I don't know.
Yes.
So what's the plan to sell the algae?
Exactly. So yeah, the plan is definitely now to sell it as food. But these are, since the production is being built, like the site is being built right now, we're in very early stages. So it's not exactly determined, let's say. But the idea is to dry it or freeze it. So process it just very simply and then sell it in either bulk to food producers or sell it for private use or for, yeah. And potentially even we're also talking about some other projects about developing products specifically with this algae. So there's some interesting projects going on, but it's all very early and it's hard to say right now how it's gonna look.
I have a question just to jump on that, like, because in cosmetics people love it.
Yeah.
Like, I know girls love it and stuff like with it and whatever, but when it comes to consumption, I don't think people don't love it, but we're just not used to it. But I mean, in Asia, it's on everything. It's fine. So how is the company doing anything to change that mindset? Because in the end, they want to make money, and so how can they change that mindset?
It's a very good question. I think first for the cosmetics parts, it's going to be hard since it's cultivated in natural seawater. So there will be, I think for cosmetics and pharma, you need to have very, very strict and like clean protocols where you can't have any contaminants. And even if those contaminants aren't dangerous or anything, I think it's going to be harder to enter that market.
And the market is already saturated in Asia and dominated. Yeah.
Exactly. But for food, I think this specific, now maybe I'm biased, but Ulva is very tasty. So it's it has quite a mild taste. So I think you can incorporate it in products without it feeling like, whoa, I'm eating seaweed, and we're not used to that in this part of the world. And we worked together with a chef who made a pesto with this Ulva, and it tasted amazing. Everyone who tried it, they were like, wow. And it's not, so I think that, I wish I had brought some, but I think incorporating it in products that we might already use to simplify like entering the market could be an idea.
there was maybe I get you take your questions.
Ah, yes. Valerie?
Would you, like, based on your observations, would you think that Ulva is, let's call it, safe for climate change scenarios?
Is Ulva safe for climate change scenario? I just repeat for people online.
That's a good question. I think it could be. It could be because it's very resilient and adaptable. So you could find maybe the right strains and work with them, and maybe change the strain depending on what happens in the climate. But of course, if temperature fluctuates a lot and starts fluctuating even more, maybe the risk of having these spontaneous zooid release in the production could increase, and that could be a big challenge. But I think just a changing environment won't be the biggest problem. It's more the fluctuation in the weather, perhaps. I would guess. Yeah. Jakob?
Yeah. You mentioned in your introduction the different salinity along the eastern coast. So I was wondering what salinity you used for your experiment, and also what implication this has. So if you want to implement a farm up there versus down where it's 20 PSU.
What about salinity?
So the salinity and what implications it has. Around Holland Bay, it's 8 PSU at the sea surface. At the sea surface. And I know that people are working with trying to get, because the Baltic Sea is very eutrophied. So it's one of the most eutrophied seas, I think, in the world. So there are initiatives mostly for restoration and conservation to cultivate seaweed to put out to accumulate nutrients further up in the Baltic Sea. So with this specific species, I'm not sure. It grows along almost up to, I would say, around 5 PSU. But then how fast it would grow and how well we could adapt these protocols, well, it remains to be seen, actually. So yeah, that somewhat answers your question. Yeah.
Thank you, Monty, for your presentation. I had a question about, like, also when Astrid mentioned that we say that this species has, like, been very used in the Asian culture and everything. Do you know if maybe, I don't know, like, how much you dig also, like, into the Asian cultivation? One, like, a part of my question is, like, haven't you tried to, like, apply their methods here? And also if there's, like, another kind of, like, in many, like, an Asian region that has similar, because maybe not the same, but like similar scenario and like similar characteristics in terms of like salinity, temperature, as you have in the Baltic.
Yes, for sure. So this specific type of cultivation is created and in Japan, so in Kochi University. So we definitely used all the protocols and all the techniques they used and tried to apply them to our specific environment. And so this is just another commercial plant, much bigger, like a larger scale, but they have the same type of hatchery techniques and the same type of growth. Because the special thing about this, well, you can see it here, they look like little clusters when they grow, and it's because by picking up the zooids, instead of them attaching to a rope, for example, or a rock that they would in the natural world, they will attach to each other, and that's the specific germline cluster technique. And then they grow out, and they have this little core in the middle, and that's the way you can cultivate them easily, because they won't like get stuck in the tank, for example. But this is all developed by Masanori Hiraoka in Japan. So for sure.
So just for my curiosity here, we're one cluster, it's different individuals clustering together.
Yes.
Okay.
So one little arm is one individual. Yeah, exactly.
I have a question about that. Sorry.
Yeah.
That you said that in the wild it's easier to find the right part of the plant to cut to reproduce. But then, do you think that will be a similar problem that you had from the lab acclimated versions? If you have them in a hatchery or in a big tank, do you think it will still be easy to find the right parts of the plant in the later phases?
Yes, I think I think maybe keeping them in this size in the lab, because the lab acclimated culture they're made from clusters as well. So they're from the production. And so here, I mean, it's easy to see where the arm ends and you can cut that apical part. But the problem is when they grow bigger, they can become, I don't know, maybe this big. And you have a bunch together, they tend to entangle. And it's very hard to, like, in theory, it should be easy to just find the core and then the the outer part. But it's quite hard to find those. So maybe keeping the clusters in a smaller size could improve that. But then they wouldn't be as mature, and I think that would also impact their reproductive properties.
Maybe a last question.
Yeah, I mean,
How long does it take them to get to a point where you can do that?
How long?
oh, how long it takes for them to grow? They grow actually very fast. The mean growth rate that we had was around 47% per day, and they can grow up to over 100% each day. So from microscopic little cluster to harvesting them, I would say maybe 3 or 4 weeks. Yeah. Let's see.
Thank you
Okay.
for our questions. Congratulations, Mathilda.
Thank you.
Microphone to Valerie. Just to make sure before you start, Valerie can just try the keyboard to make sure it works. Oh, good.
Yes.
Okay, so we're now
Nice.
moving with Valerie. So from Sweden to the Netherlands, and we are still talking about seaweed farming with Valerie. So,
Okay.
You're ready?
Okay, so that's the camera for
Absolutely.
okay, perfect, cool, thank you. Okay, good morning everyone on site and also online. I'm Valerie and welcome to my presentation on the ecological impacts of seaweed farming. When we think of farming, we normally think on agriculture on land. And before I dive straight into the main topic of my thesis, I would like to share with you a statement from the Wageningen University and Research, a Dutch leading institution on agriculture and environmental sciences. Worldwide, agriculture is the main driver for biodiversity loss. It is responsible for nearly 80% of deforestation, 70% of terrestrial, and 50% of freshwater biodiversity loss. As we expand farming to the sea, we have to ensure we don't repeat the same mistakes we made on land, which is why today's topic is so important. But first things first, why seaweed? So seaweed, scientifically also known as macroalgae, is very cool because we have loads of different applications, ranging from ingredients in food to animal feed additives to plant fertilizer, biostimulants, packaging, and so many more. Besides that, it can also contribute to environmental benefits, such as the support of marine biodiversity, the improvement of water quality, as well as potential blue carbon sequestration. So all in all, that makes it a very important contribution to the blue economy and also links it to several of the Sustainable Development Goals. So now the second question to answer in this context, why seaweed farming if we could also just harvest wild seaweed populations? Well, if we have a look at the global seaweed production, we see that Asia is the main producing country, and most of the seaweed coming from Asia is actually already cultivated. Europe, with the nascent seaweed industry in Europe, we only contribute like a little small part to it. And if we have a closer look at Europe, most of that is even coming from wild harvest, not cultivation. However, wild harvest is not a sustainable long-term option, especially not in an upscale scenario, which is why we have to focus more on cultivation. And as you can see on this map here, which shows the potential cultivation sites for cold water species, we have the better cultivation sites marked in colors from yellow to red. You can see that especially the Greater North Sea shows great potential for cultivation. And when I'm referring to cold water species, I'm mainly referring to species such as sugar kelp or winged kelp, which are brown kelp species, which are two of the most common species cultivated in Europe currently. They're normally cultivated in monocultures using the long line method, where the seaweed grows suspended in the water column by ropes, buoys, and anchors. That is considerably different to other seaweed farming methods, such as the raft or the off-bottom method. And our sort of typical seaweed farm would be a small-scale farm in a nearshore environment like this kelp farm in Norway. When it comes to the expansion of seaweed farming in Europe, one of the biggest barriers is the uncertainty about the ecological impacts of seaweed farming, which is why the objective of my thesis was to guide an ecologically sustainable scale development of seaweed farming in the Greater North Sea. And for the presentation, I focused on two of my research questions, one being, what are the ecological impacts of seaweed farming for a Greater North Sea scenario, and which have been reported, positive as negative? And how can farm siting and management influence those ecological outcomes, like either reduce negative outcomes or enhance positive ones? And I decided to conduct a systematic literature review for that, which I further complemented with backward snowballing. So I ended up with 50 papers, which were solely peer-reviewed scientific studies. And out of those 50 papers, the majority were field studies, followed by reviews. And I could also find a few modeling studies as well as qualitative case studies. And the structure, like I've extracted different information from all of those studies, starting with the possible ecological impacts for a Greater North Sea seaweed farming scenario, again, both positive as well as negative. Then I had a look at the field studies, at what impacts have been examined in Europe, the US, and Asia. And I also included recommendations on farm siting and management. And I synthesized these ones into several results sections. And to make my presentation easier to follow, I split up my discussion and placed each section after the relevant results section. So starting with the first point, what are the possible ecological impacts of a Greater North Sea seaweed farming scenario? So they're actually quite complex, and we have beneficial as well as detrimental impacts. While I could probably spend hours talking about all those impacts, I'm going to restrict myself to the two prominent ones for each side, in my opinion. So seaweed farms can contribute to the improvement of water quality. By taking up nutrients, mainly nitrogen and phosphorus, they can remove excess nutrients, potentially mitigating eutrophication. Furthermore, seaweed farms can also create habitat by providing shelter and food. And here it is important to note that it's likely that seaweed farms will attract a different biodiversity than naturally occurring ecosystems, because with seaweed being suspended in the water column and not attached to the benthos like in a natural kelp forest, they present a novel habitat, so attracting a different biodiversity than those ecosystems. On the other hand, though, with the competition for nutrients as well as light, seaweed farming can also have an impact on primary production, like impacting, for example, phytoplankton or seagrass. Furthermore, seaweed farming can also pose a risk to natural seaweed populations like kelp forests, because there could be a possible transmission of diseases from farmed to wild seaweed, as well as gene flow from farmed to wild seaweed, which could negatively impact the health of natural kelp forests, for example. So as you can see, it's a very complex picture with loads of different ecological impacts. But how does that connect to the impacts which have already been examined on field studies? There, I first had a look at field studies conducted on small-scale seaweed farms in Europe and the US. I put those two together because the scale is similar, as well as the species. It's normally the same or similar species, or like closely related species, and they're both using the longline method. And here I analyzed quite a few field studies, and which were focused on different ecological impact categories, ranging from physical-chemical parameters to biodiversity. And I furthermore analyzed if those studies had reported a positive, a not significant, or a negative impact, and looked how many studies reported such and such impact. And as you can see, although the evidence base, so the observations, are quite limited and small, none of them are significant. So I did the same for large-scale farms in Asia, where field studies have been conducted. And here you can see that the picture is a little bit more different. As you can see, we have less ecological impact categories analyzed, and the results are more mixed. We especially have also two negative impacts, which we will have a closer look at. So the first negative impact was reported seasonal hypoxia and acidification at Sancha Bay in China. And that is actually one of the world's largest seaweed farms, which has very high intensity seaweed farming year round, and it's situated in a semi-enclosed bay with restricted water exchange. That's important to know because there the situation was that seaweed litter, so broken off seaweed fragments, accumulated on the sea floor, which led to a very localized deposition and therefore also a localized decomposition, which would significantly reduced the oxygen in the water, leading to those hypoxic conditions. And the other example was a reported seagrass biomass decline at an off-bottom farm in Indonesia. And as you can remember from the introduction maybe, off-bottom farming, which happens directly on the benthos, on the sea floor, is considerably different to what we would normally use in Europe with the longline farming. Therefore, it can also explain those aggravated effects on the benthos, like the seagrass. So to conclude, we have limited ecological impacts in Europe and the US on small-scale farms, while in Asia, the picture is a little bit more mixed. And that generally corresponds to literature also saying that the ecological impacts of seaweed farming at current European scales are negligible. But what we could note is also that we were working with a very limited evidence base. There were only few field studies available in English, which might be a point because, for example, a study done by Spilias et al., who did a similar review, they also included field studies in Japanese, Chinese, and Korean, so the languages of some of the main producing countries. And he could identify way more field studies and observations. Another thing we could note was that there was a high unevenness in examined impact categories. So most impact categories were actually on water quality, and there were actually no reported observations on, for example, marine mammals or seabirds, at least none that I could find in my literature research. And that was something which the previous study I just mentioned, Spilias et al., could also confirm. Most of their observations were on water quality as well, because it's easier to access or assess than marine mammals, for example. Furthermore, to consider is that it could be more likely that positive impacts are rather reported than negative ones, which we have to consider as well. So there might be a bias. But even with those limited, even with all of those limitations, we could still see that there is an influence on the ecological impacts when it comes to farming method and especially farming scale. And that brings us to the concept of ecological carrying capacity, which is the threshold beyond which aquaculture results in unacceptable environmental challenges. And you can kind of like imagine it like this. When we're moving from a smaller scale farming operations beyond the ecological carrying capacity to a larger scale, the beneficial impacts could result in more detrimental impacts, and sometimes those beneficial impacts can also become detrimental impacts, like it was the case for the example of Sancha Bay, because on smaller scales, seaweed litter, so like the broken off seaweed fragments, could also be a food source for marine biodiversity. But since the scale of this farming might have been a little bit too extensive, this can result in those hypoxic conditions they have reported. But so far, there is no standardized framework for assessing carrying capacity for seaweed culture at the moment. However, many studies suggest using coupled physical biological models, which can give us insight into the nutrient budget as well as potential impacts on phytoplankton and other trophic levels. And the ecological carrying capacity of a site is highly connected to the local environmental conditions. So it's extremely important to choose a suitable farming site, which brings us to the recommendations for this part. So we definitely want to place a seaweed farm in an area where we have enough nutrients, mainly nitrogen and phosphorus. They could come from natural sources or even better from anthropogenic sources, so we can mitigate eutrophication in the beginning. Besides that, we also want to have a moderate to high current flow, not only to ensure nutrient replenishment, but also to dissipate any negative impacts. That is something we definitely want to have. What we want to avoid are sensitive areas. And with that, I mean, for example, protected seagrass or merbeds, or even like known areas of migration or foraging for marine protected marine mammals. because at the current evidence base, we don't know enough to really be sure about the ecological impact of seaweed farming on those impact categories. Instead, we should place seaweed farms on sandy bottom or even degraded areas, because there it is likely that seaweed farms rather contribute to ecosystem services than reduce them. Also important are farm management recommendations. These include primarily only using native seaweed species which haven't been genetically modified yet to limit any impact on natural seaweed populations. In the future, there might be a solution to it, but to my knowledge, that is not yet commercially established to be used nowadays on bigger scales. Secondly, it's also important to implement a rigorous biosecurity management plan, which especially focuses on pathogens, invasive species, and pests. And another good recommendation was to diversify the culture, which means not only farming different seaweed species with each other, but also incorporating seaweed into integrated multitrophic aquaculture, IMTAP, so farming seaweed among bivalves like oysters or even finfish, because that has been shown to have not only environmental but also economic benefits. But all of these are mainly suggestions, and very few have been actually backed up by empirical evidence. And the scarcity of the field studies as well have shown us that there really is a lack of standardized ecological monitoring. And here would be important to provide or to do asymmetrical BACI study designs. And BACI means actually before impact, after impact. So we're having a look at the seaweed side before the implementation, during the operations, and after. While the asymmetrical part stands for having multiple control sites to accommodate for natural variability. So here we can have a look at more localized effects, but it would also be important to combine the field measurements with modeling studies, as I've mentioned before. So I'm already at the end, so if I want you to take away 3 key messages, it would be that the current impacts of seaweed farming in Europe are actually limited, but the evidence base is limited as well. Secondly, it would be that the local context can have a high influence, but that as well is not backed up by evidence yet. And thirdly, it would be for the future expansion of seed farming, we should apply a precautionary and evidence-based approach backed up with long-term ecological monitoring studies, as well as an adaptive management. That was my presentation. Thank you for your attention.
Thank you, Valerie. I would like to thank also Yves Brouwers, your supervisor at the European Seaweed Association, so in the Netherlands. We're going to start the question with Juliette online, going on with Robin after.
Hi, Valerie. Thank you for your presentation. I wanted to ask, let's start with your studies on Asia, when you were comparing the few studies you found for Europe and then the impact you found for Asia. So you talked the 2 negative ones were using another technique, if I'm not wrong, than the one they want to. Yeah, one of them. And what about the other ones, the ones with even positive impact or no impact? Was it similar than the one they want to use in Norway, or is it a mix all the time?
So not every, those were in total 10 field studies from Asia I have included, and not every one of them did really specify the cultivation method. But the ones who did, except for the one in Indonesia with the off-bottom farming, the other ones which did specify it, they were using long line cultivation method, which is similar to what we are doing and what we would expand in Europe.
Okay, and so you talked about marine mammals, but there's no study on them, and that we can't know what would be the impact. But in your opinion, what could be an impact? Because are the farms usually based very close to the shore, or are they very far away? And I'm interested in what you think what could be an impact with the different methods, or at least the method that's most prone to have in Europe.
So I think there's a lot of unclarity about the impacts on marine mammals, and I think the only known case was a reported fatality for dugong on a seaweed farm in the Philippines. But I think I only found that out when I was comparing the review, which I kind of like took as one to confirm the findings of my studies. So I actually I could not find this report in the literature I have accessed in the systematic literature review. But I think they're, like especially with marine mammals, like they could be attracted to the farm because the farm would also attract fish. So they could use it as a hunting ground, but at the same time, the farm management equipment could also be like pose obstacles to their hunting methods, like if they're trying to school up fish of like prey fish. And another risk we have to consider would be the potential risk of the entanglement, which could lead to fatalities. Yeah, and it's like really difficult because they have not been, because we don't know in which direction the impact would rather lean. But I think since they're protected, I would rather apply a precautionary approach and say it's rather negative, unless we can prove that it's not.
And using what you suggest to do, like a BACI, then it would allow probably to identify areas where there is marine mammals, maybe avoid those areas to install, right?
Yeah, I think like the like especially with marine mammals, because like they have lower population densities, and then they're also like they're migratory as well sometimes. I think to examine the impact with BACI studies on marine mammals, you would really have to do a long-term study. So that would quite take a few years and a lot of effort to get good results with it, which is one of the reasons why I think they have not, there is so little known about the impacts on them. It's difficult.
And so the study you mentioned where he managed to understand at least the papers in Korean, Japanese or Chinese, he was resuming, you could take the data from his study after he analyzed them, or was it still hard for you to get the information from those papers?
I think he was doing, so like for the understanding, I could not identify the study during the literature, the systematic literature research review, because it, I would say, maybe escaped my predefined search terms, to put it in that way. And like from the data he could provide, it was also more of a review of what could be likely, for example, the habitat provision for marine biodiversity, and what could be unlikely to have positive or negative impact. So it was a little bit tricky to take the data, also because he was since they focused on as well as on Chinese, Japanese, and Korean studies. I also don't know what conditions were prevalent at the seaweed farms, if it was similar to European case studies. But because I didn't have access to all those field studies, it was difficult to take those results to include them into my study as well.
Okay. And just curiosity, so you based your literature review on Scopus. Did you think of going more deep as well, using other like Web of Science, Google Scholar, to make sure you were not missing papers, or you like, I'm curious to know.
Yeah, so I checked Web of Science as well, but Scopus could actually provide me more results. And to kind of like, because I know like there are limitations to systematic literature reviews, that's why I complemented it with backward snowballing, so that I could identify additional literature when checking the references of the studies I've already identified. That was kind of like my measure to work against those limitations. But I checked Web of Science as well, but there was more on Scopus. And
Okay, thank you, Valerie.
Thank you, Juliette. So we can go to you, Robin, and finish with thesis.
Thank you for your presentation on seaweed again. So it's really something I'm not very familiar with. I will not be able to remember the term that you use, but I hope you can understand my question. What could be the issues related to the use of sterile sporo something?
So you mean like the issues of as in like potential risks? Okay.
Yes, of using these.
Since didn't really have time to explain stereosporophytes, it would be, so there are sporophytes which are actually not able to reproduce in nature. So we wouldn't have any risk of gene flow from, or like a very limited risk of gene flow from farm to wild seed, for your understanding. And potential risk of that could be that although there is a quite, as I said, like they can reduce the risk of gene flow, the risk still persists because there could be mutations which could reverse the sterility. So there would be sterile, but we would be thinking there would not be sterile. And then we could have the gene flow again, and that would be bad in case we would start to genetically modify the strains of seaweed we're using. For example, cultivating them or like breeding them more in that sense that they are good for cultivation, but then they might have a lower genetic diversity. And if we have that gene flow from farm to wild seed with a lower genetic diversity, then wild seaweed could also have a lower genetic diversity, which would make it more susceptible to diseases, pests, and environmental changes. So we would weaken their health. That is the risk with stereosporophytes. But from the literature I've read, it's a limited risk.
And would there be any commercial or economical risk of having, I mean, I don't know who can produce this sterile sporo something again? Yeah. I mean, I'm thinking of what happens in the agriculture on land. That's basically you have a monopoly of big companies that just produce these sterile things, and so it's they keep the control on the seed and on the production of this. I don't know how it. I mean, I don't know if that could be an issue with sea farms also, or if anyone can produce their own sterile sporo or something.
Can you rephrase your question?
Who can produce? Yeah.
Oh, go ahead, Robert. You can rephrase.
Who can produce? How is it difficult to produce sterile sporo things?
Sporophytes.
So I have no practical experience in that, but I was lucky enough to visit a seaweed nursery and a company who was also producing. It's called producing seeds, which I could ask. So they told me it's not commercially established yet, and it would be companies like those who could produce them. There are two ways to how to produce them, but it's not as easy as it is, or as it should be for commercial application.
That's why Robert, I think, was referring to what's going on currently on land with agriculture, knowing that you have a few companies who develop the skills to do that, and so we keep the monopoly, and so creating sometimes inequalities. So what do you think will be, they will know what, once a few companies acquire the skills to do that, what can we expect? What do you think?
I hope not the same as on land, where there's one monopoly, providing the, let's call it genetically modified seeds even, which are very good for cultivation. I hope that won't be the case. And I think therefore it's also important to establish, that's what many papers also mentioned, that we should establish many national seed banks, not only to preserve the wild seaweed genetics, but also to breed the strains we want to have if it's safe to breed them. And I hope that if we have many national institutions, we can avoid the same scenario as we have with terrestrial agriculture. But of course, I'm not the one deciding it. So,
can I ask the last question?
Of course.
Yeah. So in Europe, there is very limited seaweed farming, and most of it is harvesting, from what I understood. Why is that? Is there no interest in company? Is there no market? Is there? Why is the Asians so good at it and us so? I don't know. Maybe we just don't do it. I mean, why is there so few in Europe?
So as Mathilda has already mentioned, in Asia they have been practicing that for a long time, and especially seaweed is used as a human food source, so it's rather treated as a commodity. while in Europe, seaweed also has quite a bit of history, but more the wild harvest, especially like in countries such as Ireland, Scotland, Norway, France. And cultivation is just like picking up recently, and there's still loads of barriers for the expansion of the cultivation. And as I said, one of them was the uncertainty about ecological impacts, but there's also like the market is not as developed for seaweed here in Europe as it is for Asia. And then a second really important reason is also the production costs are way too high. So at the moment, I think it's the low or the cheapest ones are actually imports from Asia, followed by European wild harvested seaweed, and then the most pricey one is actually cultivated seaweed. But I think that also depends is connected to the current seaweed farming scales, because small scale farming operations generally cost more than, for example, larger scale farming operations because they can reduce the costs. So it's a little bit tricky because they're connected, those reasons. And like there are a few other issues as well, but I think I would go into too much detail.
Maybe you will continue to answer some questions
Yes.
also. So sorry, I'm not from Mathilda, of course.
I like your introduction about kind of what I understood of it, getting ahead of the curve, like kind of learning from terrestrial farming and things like that. But obviously, there's many things that have gone wrong in terrestrial farming, and it will heavily influence the kelp farming industry. I'm interested in your search, and Julia asked one of the questions on Scopus. But what were the search terms that you used for the search itself? Mainly because I completely understand it's difficult to get the literature from Asia. A lot of them. Did you just look at peer-reviewed papers?
Yes.
Yeah. So that excludes a lot in Asia.
Yes.
Peer review was 1970s. A lot of reports are technical reports, so the data will be there, and you need to have Japanese, Chinese friends to translate them.
Yeah.
So I'm just interested in what were the search terms that you used for Scopus?
So I've unfortunately forgotten to include a slide on the annex for that, but I remember them. So I was looking for seaweed, macroalgae, and kelp that like connected with the ore connection, and then with the end connection, I had farming, mariculture, agriculture to kind of like get all of those three. They were also connected with an ore connection, and then another end connection with environmental impacts, impacts in general, positive impacts, negative impacts. So quite a lot of that. But for example, what I could have included, what I was thinking afterwards, were also like environmental risks, environmental benefits.
Impact assessment.
Yes. That's something I could have included, but on the other side, also kind of had to narrow it down because I did not have time to read, I don't know, hundreds of papers.
I understand. Yeah. No. I think the search terms were were a good start. But one question I have about the results of it, especially in the Asian context, is what about cumulative impacts? You know, there's never really one impact, especially when it comes to farming. So how did you handle the cumulative impacts of?
So I would say I think it's a really good question. Like especially like with cumulative impacts, what I've read about literature was that they could, like, it's tricky to determine cumulative impacts if we're even not sure about the impacts of one specific seaweed farming site, as you could see. But I think the closest approach was doing it with modeling studies. For example, there was one modeling study where they had placed different farms in seaweed farms in Europe, and they were modeling how the nutrient reduction would affect the phytoplankton population. And there they could also see, like, depending on how close they placed the farms, with which intensity that would have cumulative effects. So I think like those modeling studies are the best approach to have a look at those cumulative effects. But it's difficult for other impacts, I would say, because
I think on your graph it would have just been nice to have another category of cumulative impacts.
Which graphic do you mean?
Slide 13 maybe.
13.
Yeah, you don't need to go back, it's fine.
Okay.
But just to have that, because it can be its own And you know, if you know you're using this as a report and directing whoever to this farming, to know that cumulative impacts are one of the biggest kind of things, because life is different in Asia. I would say like in the cumulative impacts are felt from land to sea, sea to land, on local people, you know, on a large scale, and we don't have that here yet. For sure. So it would have been nice to have that, but you did a lot already. So, and my final question is about the aim of the company, or whatever it is,
European Seaweed Association.
European Seaweed Association. Are they focused on because food security, or is it for kind of carbon sequestration? What is the main kind of driver of the farming? Would you say?
So you mean like the main objective of the
is it for to sell this product?
It's really like they're really focusing on the market development, which is like, as I said, like there are many barriers and problems connected to each other, but they're really focusing on the market development, and the main aim with pushing for seed cultivation is to realize it within ecological boundaries of the planet. So like really ecological sustainability. And then like with because you mentioned, I think, blue carbon or food security, they're not specific.
No, you mentioned it in your abstract.
Okay.
Yeah.
Okay. The thing is, like with food security, I think we are like seaweed can contribute to food, but also I think in Europe we also have big potential for the other applications of seaweed farming, such as, for example, biostimulants, which are also good for terrestrial agriculture. Again, kind of like closing the loop.
Yeah.
And blue.
Yeah. Okay. And my final question is the trajectories of these two species that you focused on.
Yes.
What are their trajectories? Are they in decline, recovery, stable?
You mean the production, or?
No, just natural populations.
Natural populations. I have to admit I did not research that, so I did not research that.
That's fine.
But I think because it was not in the scope, I think what was in the scope was like limiting or finding out what could have an impact and how to limit that impact.
Yeah. Okay.
And I think then like those natural populations, they're also subject to many other threats.
Yeah. Fantastic. Yeah, I really enjoyed it. Thanks.
We have a couple of minutes for your questions.
Yes. Oh, hey. Mathilda.
Thank you, Valerie. It was really interesting. And I was wondering, in the beginning where you have the map of suitable places for seaweed cultivation, it was, of course, interesting to me because the Baltic Sea was very, very low. But was that specifically for Saccharina and Alaria, or was it for all kinds of seaweeds? What was it explaining?
It was like the map I showed in the beginning was for cold water species, like those large brown kelp species, for example. And then the study was really interesting because they also focused on intermediate species, which are green species. And I think I also included a nice slide in the appendix, which was kind of like having a look at the different limitations of seaweed farming in different areas around Europe. But even like also for the intermediate species, the Baltic Sea didn't look great. Sorry. But I think it's because the salinity, as you've mentioned, is a very deciding factor. I'm just going like Andrea.
Thank you, Valerie. Really nice presentation. I had an interesting question. Yeah.
Good for you.
Like this connection in my head.
In a few words, Andrea.
Can you go to
In a few words.
Can you go to the part where you show the positive impact?
Yeah.
The two ones that you selected. You mentioned that you found habitat creation and like the noble habitat creation as the positive
Yes.
like a beneficial aspect. Wouldn't that also could be considered as a negative impact if you create new habitat and that which could drive to invasive species maybe, or change in community in the habitat?
Yes, good observation. Because like the studies also emphasized that not every increase in biodiversity can be seen as a positive increase, especially if it's like invasive species, because like the artificial equipment we have, like the ropes, buoys, anchors, they could be a good stepping stone for invasive species. And I'm not only referring like to invasive seaweed species, but as well, of course, but other invasive species like epibionts, which are not native. So it could also be a negative impact. It is kind of like two sides of the same coin, but I chose to, as I said, if I would have more time, I would have listed them all, but good observation.
Two last questions.
Corinna, and then after Lapo.
You mentioned this one study or farm in Indonesia that they grow them in the bottom, and I'm wondering why is the long line and the way you create habitat that I guess it attracts more fish and megafauna instead of promoting the benthic ecosystem that is the natural way for kelp to grow. Why? And if I can add one thing, the Saccharina latissima, for at least speaking for Spain, I know that it's not endangered and it's the species that it grows everywhere, and that's the one they farm. And there they grow it in the rocks, from what I know.
Do they grow it in the rocks, or is it naturally growing in the rocks and they just harvest it?
Yeah, I think they're recultivating it, so they're not just harvesting wild.
Or like restoration?
Sure.
Okay.
Just wondering if you got more into specific contexts in Europe, and why not the benthic growing?
In a few words, if you want a question from Lapo. In a few words.
No, that's okay. What was the question? Okay. Wait, I'm gonna answer your question. So the farming really depends on the species we're farming. Like for the quite largely growing brown kelp species, the longline method makes more sense. And like what they use for the off-bottom method, are the species like red seaweed species such as Cappaficus or Choima, which are growing like more bushy and not like those brown kelp species. And like, but we don't have the suitable conditions here in Europe to grow most of them, which is why they have in Indonesia and other parts of Asia, and there they grow them with those off-bottom methods. So it's really species dependent.
Maybe a short question and answer from Lapo and so forth.
Okay, sorry.
Yeah, thank you. Super interesting presentation. When I read the paper, you talked about ILPA, which is indeed, as you say, to have a positive ecological and economic impacts if done smartly. But my question was more, if based on your literature review, did you find any studies, or if not, did you generally form any opinion on whether algae farming could be used to absorb some of the negative impacts of aquaculture more in general, especially also on intensive aquaculture?
So,
if, of course, integrated properly, adapted with practices.
The question is about seaweed culture as absorbing negative impacts of fish farming.
Yeah, there are like quite a few field studies who have solely focused on the nutrient absorption. And they have field studies with differing methods, so it's difficult to compare them, but they all could identify that that seaweed would take up nutrients, because they would, for example, check the content of the seaweed, look how much nitrogen and how much phosphorus is in there, and could kind of calculate, okay, the seaweed took up so and so much of the nutrients which would be produced by the fish farms, especially in combination with bivalves. So if you have those two together, they're quite powerful. Yes.
Thank you, and congratulations, Valerie. And before we go for a break, just you had Valerie and Mathilda a few hi, so from Emily on side, but also Mathilda, I think it concerns you. Great presentation, and some people are very happy to choose you to work with them, and they say welcome back. So I guess it's good news. Your grandma, oh, nice. Also, so hi from North Sea Farm something. This is for you, I think, Valerie. Also, great work. and more high. Oh, picture is nice. Thank you. So we have a short break, but I wanted you to have some question time. We are back here at 11:10. So it's in less than 15 minutes. Okay, some for the jury and people online. Good. So we are all back here. Yes. And this is the second part, the last part of the last session. So in here, we are going to go with Lena Merz and Jakob Roeckl. We are still talking about sustainable ocean innovation. Robin, be ready. You will start the questions because this time we talk about fish and fisheries. Yeah. Okay. You can go on the next and come back. Yeah, sometimes it's if you're all ready, Lena?
Yeah. Okay.
Okay, you can go.
Did you know that there is a fishing technique that is so important it captures 66% of all tuna worldwide? Well, at the same time, it also captures 20,000 shark individuals as bycatch every year only in the Eastern Pacific Ocean. So my thesis was all about finding a balance between these two interests, the tuna per se fishing industry and the reduction of shark bycatch around Baja California Sur in Mexico. I wrote my thesis under the supervision of Frida Lara Lizardi, and I made a spatiotemporal analysis that can be used for local adaptive fisheries management. But what are tuna purseiners? So we're talking about these large industrial vessels, and they locate tuna schools, and then they put a large net that can be up to 2 kilometers long and 150 meters deep around these tuna schools. They close the net on the bottom, they capture all the fish and retrieve it back to the boat. There are different types of locating the tuna schools, and these define different strategies and set types. A set is this whole procedure of finding one school and capturing it. In my study, 2 of them were most relevant. The first one is called unassociated set. This is when they capture free schooling tunas. So they use either helicopters or observation platforms to find these schools, and then they capture them. The second set type is called dolphin-associated sets. So especially in the Eastern Pacific Ocean, we know that often yellowfin tuna, they're associated with pods of dolphins. So instead of finding the tunas, they can target the dolphins and let the dolphins escape before they retrieve all the tuna back to the boat. The problem is that the selectivity of this technique really depends on the set type and also on the operational characteristics of the fishing. So if the selectivity is low, this can lead to a high rate of non-target catch, and this is bycatch. In my study, I focused on shark bycatch because they're important to keep the ecosystem in an equilibrium, and they have life history traits such as long lifespans, low fecundity, and late sexual maturity, so they are susceptible to such pressures. To look at the interaction between fisheries and non-target species, it's especially interesting to focus on areas of very high productivity, where different species across the food web share their habitat. And Baja California Sur in Mexico, on the west coast of Mexico, is one such area. So I included the waters of the Gulf of California here in blue, and then also the waters of the open Pacific Ocean until the Mexican EEZ in orange. And as I said, we're located on the west coast of Mexico, and we are within the convention area of the Inter-American Tropical Tuna Commission, the IATTC, and their role is to manage tuna fisheries to make it as sustainable as possible. So what was my research question? In my thesis, I wanted to answer where and when do the patterns across space and time of tuna catch and shark bycatch rates indicate recurrent fishing inefficiency. And I will explain what recurrent fishing inefficiency means in a bit. But first, let's see how I answered this question. So I had 4 different research goals. The first one was to characterize the seasonal tuna per se fishing dynamics. So I wanted to know who is fishing and where are they fishing. So for every month of the year, I made a map of the fishing effort. Then once I knew who is fishing and when they are fishing, I wanted to know what are they fishing. So again, I mapped the distribution of tuna catch and shark bycatch rates divided by the different set types and the different species. After doing that, I could move on to my inefficiency analysis. And fishing inefficiency simply means that in the same place and at the same time, we have a low tuna catch rate that coincides with a high shark bycatch rate. So neither for the fishing industry nor for the conservation of sharks that is very efficient. After identifying these areas, I also wanted to know what would be the implications of management. So I calculated how much tuna catch would be reduced or would be lost in case of closing these areas and how much shark bycatch could be avoided. And for the presentation today, I'm focusing on goal 3 and 4. So what methods did I use? First of all, the data set that I used is from the IATTC, the Inter-American Tropical Tuna Commission, because they do mandate a 100% observer coverage on all large tuna vessels. So we have these observers on board. They write down all the tuna catch that is happening, but also all the bycatch. And then they publish this data. I was working on a 1 degree spatial resolution and on a monthly temporal resolution. And my study period was from 2010 to 2024. So in the end, I had around 18,000 sets to analyze. Then I made different categories. The first one is the catch category. Here, I included all the species that contributed more than 1% of the total catch. So overall, we had 240,000 tons of tuna across the study period. And most of them were yellowfin tuna, followed by skipjack tuna, black skipjack, and Eastern Pacific and striped bonitos. For the bycatch, instead of just making one bycatch group, I divided into different focal species. And to select the focal species, I looked at the bycatch rates to include the ones that were mostly bycaught, but also at the IUCN red list status to make sure that it's actually important for and relevant for conservation. So we had 3,500 individuals that were caught, and I focused on the bycatch of silky sharks. These were the most bycaught species. Then thresher sharks. Here, the data set does not distinguish between the different species, so it's just one group. And scalloped hammerheads. So silky sharks are vulnerable. Thresher sharks are between vulnerable and endangered, and the scalloped hammerhead is critically endangered. Then, instead of only looking at the total catches, I wanted to calculate catch rates and normalize the data. So I calculated the catch per unit effort, which is the tons of tuna divided by the number of sets, and bycatch per unit effort, BPUE, which is the number of shark individuals divided by the number of sets. And then for my efficiency analysis, I could compare these rates to monthly averages. So the monthly average is simply the average of, for example, the CPUE or the BPUE of the whole study area and the whole study period for a specific month. So in the end, for every January, February, I had an average CPUE and average BPUEs. And then if the CPUE was below that average in a specific cell and a specific month, and at the same time the BPUE was above this average, it was classified as inefficient. So in simple words, if we have a lot of shark bycatch and not a lot of tuna catch in the same cell and the same month, it's inefficient. And then I distinguished between single events that happened only once throughout the study period and then recurrent events that happened at least twice or more. And then I can make maps that look a bit like this. So we have the gray areas. These were not of my interest, and they were classified as efficient. And then yellow areas are inefficient, but only single events. And the red cells are the cells of my interest because these are the recurrently inefficient areas. So I told you we have these two different set types. I made the analysis for both of them, but then for this presentation, I'm focusing on the dolphin-associated sets. So let's look at the results. Here you can see one overview map for the dolphin-associated sets and all the shark focal species combined. And what can we see? So we can see that there are 9 recurrently inefficient cells. And my temporal analysis showed that this recurrency happened between March and August, so never during the winter months. And what does this tell us? we can see that this inefficiency is spatially very concentrated to the southern study area, but also more to coastal areas rather than here the fully pelagic offshore areas. And comparing this to my literature review, I found that these areas actually overlap with ecologically sensitive and important areas, because these are shark nursery areas and also tuna spawning grounds. So we have this shared habitat down there. So if you go more into detail for every species, you can see the results for silky sharks, for thresher sharks, and for scalloped hammerheads. And we can see that most of the recurrent inefficiency is driven by silky sharks. We have all the 9 recurrently inefficient cells there. And then for thresher sharks, there are 2 recurrently inefficient cells, and they are here located at the entrance of the Gulf of California. And also the single events, they show a very clear pattern within the Gulf of California in the coastal areas. Then for scalloped hammerheads, there was actually no recurrent inefficiency and only 2 single events, and they were also located at the entrance of the Gulf of California. So we'll discuss what that means for every species in a bit, but then first let me show you what the management implications would be. So as I told you, I calculated how much tuna catch and how much shark bycatch was caught throughout the study period in these 9 cells during the months of inefficiency. And these are the results. So by avoiding these areas, we could have reduced shark bycatch by 17.2%, while only 7.9% of the tuna production would have been lost. So this tells us that proportionally, sharks would have benefited by this management approach twice as much as the tuna industry would have been affected. Looking at the different species again, we see that silky sharks and thresher sharks, they would have had a reduction of around 17–18%, whereas scalloped hammerheads would have been reduced by around 10%. So all the species exceeded the 7.9% of tuna production loss. If we then look at absolute numbers, we see different results for the different species. So for silky sharks, in total, 192 sharks would have been avoided to be ended up as bycatch. Whereas thresher sharks, it was 12 individuals, and for scalloped hammerheads, it would have been 1. So let's look at the results for all these species. First of all, I want to highlight that it is hard to contextualize these absolute numbers in the absence of a good stock assessment, which is the case in the Gulf of California. So if we don't know how many individuals are there, we can't really tell is 100 or 10 a lot or not that much. But we still have some interesting insights. So you saw for silky sharks, they drove most of the recurrent inefficiency. They also had the highest number of bycatch. And in the literature, it is a common problem that silky sharks end up as bycatch. So I think this management approach would be a good approach and a feasible approach for silky shark conservation. For thresher sharks, we saw that there were fewer hotspots. However, this is very interesting because we know that thresher sharks are very, very migratory species, and it is hard to protect migratory species. So if we already know that they come to these coastal areas in the Gulf of California for breeding and nursing their young in certain months, and at the same time the fishing industry is not efficient in these areas, I think it's a valuable insight for future management. Then going on with scalloped hammerheads, there was actually no recurrent fishing inefficiency, as you saw. However, this does not mean that they are not of conservation concern. It's actually quite the opposite. We know that in the Gulf of California, the populations have fully collapsed, and they were reduced since the 1970s by around 97 to even 100%. So nowadays, every single interaction matters and probably decides if this population will survive or not. So maybe this management approach is not the most suitable one for populations that are already so depleted, but they should still be considered for future management. Overall, I would say that my results are not necessarily a call for immediate closure or immediate management, but they did show us where the priority areas are for the future, how they overlap with ecologically sensitive areas. And then I would say that we need more social and economic and also ecological assessment to really understand what these results mean and how we can implement this in management. So as a conclusion, we saw that the recurrent fishing inefficiency in my study area is spatially very concentrated, and it overlaps with ecologically sensitive areas. Then after this analysis, we know that dolphin-associated sets and silky sharks drive most of the inefficiency. And by managing these areas, we could reduce shark bycatch by 17.2% while losing 7.9% of tuna production. For future research, there would be some interesting impact aspects to include in the analysis. First of all, I would like to model the fleet redistribution in case of management. So if some areas get closed to fisheries, where would the fleet go instead? And how would that impact bycatch and catch rates? Secondly, I would like to zoom out and look at the bigger picture, because tuna perceiners are only one of the pressures. And I would like to make an overlapping analysis of different pressures to look at the cumulative impacts. And then I would like to implement also, or include also, more factors, environmental factors and social and operational factors, to really understand what drives the fishing effort and also these inefficiency hotspots. So I want to thank Frida for your great supervision, and all the team in Mexico and Orcas for the collaborations, and Guillermo for being a great advisor, and all of you for listening and being here today.
Thank you, Lena. So I would like also to thank Frida Lizardi, your supervisor, and Orcas Basin in Mexico. We'll open the floor to Robin, who is this time, I think, not saying that he doesn't know anything about fisheries and fish, so we're good.
Yes, probably, I mean, hopefully relevant questions. I don't know, we'll see. I actually read your report, so I know a bit more what you've done than what you've presented here, but I will keep my object question for the last, because you didn't present it at all. So first, it was very interesting. I think that's a good approach, and the examples where it works relatively well, in particular for swordfish in the Pacific. Operationally, what spatiotemporal resolution do you think would be needed to make it, to make it operational in a perfect world and in the real world?
I mean, I talked to some people that work in management and the IUCN, and they said that a 1 degree resolution is actually a good resolution for management. However, I think working on such a small study area, it would be interesting to work on a finer scale as well. Especially now that we know where these priority areas are, I would like to go down to maybe a 0.5 or even 0.1 resolution, and really understand what would be the detailed differences. Temporally, I think a monthly resolution is probably not bad, because then also if you go into too many details, it doesn't necessarily make it easier for management. So yeah, it's always interesting for analysis to get more details, but then for management, I would say that you have to take a step back again and make it, more feasible for,
Okay, thank you.
That's what I would say.
What do you think would happen if you consider median CPUE and BPUE compared to the mean that you have used throughout your analysis?
If I worked with the median CPUE, so you would say for every cell?
Okay, I'm gonna drive you a bit. You have events with high levels of bycatch.
Yeah.
So that means that your average over the months is gonna be relatively high. But if you have a single event with very high level of bycatch throughout your months, your median is gonna be 0, while your mean is gonna be, let's say, 50 or something like that.
I mean, it would probably change the efficiency classification either a lot or because I looked at the outliers and the box plot analysis. So I do have some outliers, and sometimes some of these outliers might have a big impact. So I think it will probably change the classification of the inefficiency, and sometimes, depending if you look at CPUE or BPUE, it would either not be classified as inefficient or more cells could be included. But yeah, I'm trying to think it through.
I don't have the answer. I just think that it might change some of the results. And so I really suggest that you think about it if you or other people work on it to do both, because sometimes the mean is representative. In this case, when you have rare exceptional events of bycatch, the mean is very unlikely to be the most representative metric that you want to use.
Yeah, it would be interesting to include that. What I try to do, at least by having the threshold of at least 2 events, I try to eliminate all these outliers of single events. But I agree that it would be interesting to see how different would be the results and what would be the impact. Yeah, I find it hard right now to foresee the results, but it would be interesting to compare them. I agree.
I mean, if you can foresee them without computing them, you are a genius. One more, two more questions if I can. For the fishing effort estimation, you didn't present it more in detail in the presentation, which I understand. What data did you use? Because it seems like you use different categories of vessels, including the class 6 vessels that you end up using, but also smaller ones. Is that
4 to 6, class 4 to 6? Yeah. So I
use class 6 for the bycatch.
I think the mandatory observer data is mostly focused on class 6, but then they also have observers on class 4 and 5. So I just included all the data that I had access to, which is between class 4 and 6. But then they always use the same for the fishing effort. I always use the same unit, which is number of sets. In the beginning, I also wanted to compare it to other types of fishing. Like I want to compare it to the fishing hours according to AIS and VMS data. But then it was not that easy to compare these different metrics. So I focused on fishing sets as a unit and stated that, yeah.
What was the fishing effort in the inefficient cells? I mean, the cells that you detected as inefficient, because you said it reduces by 17% the bycatch of sharks and 8% the bycatch of tuna. But what was the effort in these cells compared to the other ones?
That's a good question. You mean a number of sets? Or
the fishing effort.
I didn't calculate that. I only calculated how much tuna catch would have been caught. And I can tell you what a lot of annexes, but I can tell you what the what the CPU was in these cells. In these months of inefficiency, but then I don't know what the CPUE was in these areas. But if we look at the dolphin-associated sets on the left, and then the months of inefficiency, which was roughly between, let's say, here, like the summer months, then you can see that the tuna CPUE was around 7 to 9 tons of tuna per set. But then it would be interesting to see how many sets were actually deployed in these areas. But I know, looking at the fishing effort maps during these months, we do now know that in these months the fishing effort is high in these areas of inefficiency. But I can't tell you an exact number. Yeah.
Okay. Can I ask a last question, or I keep it if we have time at the end? Yes. Okay. So you had actually 3 categories in the set: the dolphin ones, the dolphin-associated ones, the free swimming, and the floating object associated sets, which actually showed very different results to the 2 other ones. Why didn't you include them in your analysis, even with the precautionary approach of saying, okay, this has to be? You had less strengths, but you still had data.
I get your point. So maybe for you guys, there's also a third set type, which is called object-associated sets. So they build these fish aggregating devices where a lot of species get attracted. And usually these are the ones that are known to have a high bycatch rate. And in my study area, there were 79 floating object sets throughout the study period of 15 years. And in these 79 sets, there was a high bycatch rate, like way higher than the ones that I had in my analysis. But I wanted to make it more relevant to the reality of this area. So if I already know that, because if we compare the numbers, I think I do have them here. But yeah, here, so we had 8,000 dolphin sets, 10,000 unassociated sets, and 79 floating object sets. So I felt like an easy management approach would be just to ban the object sets because they're not really used anyways. So I wanted to make it more, tailor it more to the reality of the area and just focus on the ones that are used.
But you didn't say that. That it could be a way to ban it. And the thing is that, for example, I mean, I don't know if it's the case, but if there's only 80 sets, but all of them lead to high bycatch levels of hammerhead, then they have a significant impact, much more than the other types of sets. So I think you may not want to discard them completely, just saying we don't have enough data, because they may have a different effect to the other sets.
Yeah, that's true. I mean, I explored it. It was, you can see the years here where it happened from, it was more in 2010 and the last ones here in 2019 for object sets. And I did an exploratory analysis and I saw that it was mostly silky sharks. Sometimes they had a single set and more than 100 silky sharks in one set. So I did analyze it just in my exploratory analysis, and then I felt like I already had a lot of results, so I tried to narrow it down again for the thesis. But yeah. I agree that it could have been interesting to include it as well in the text.
Thank you. Thank you, Robin. Juliette, it's to you now.
Okay, thank you, very interesting. So I'm not at all an expert as Robin, but so you decided to focus only on the one method here, on the dolphin one. And I was just wondering because I haven't read your report, what was the results for the other method? And yeah, why did you choose to focus on the dolphin one?
So for the other method, you can see the results here. On the left side, there's the dolphin-associated sets, what you just saw. And on the right side, these are the unassociated sets. And we can see that there's only one recurrently inefficient cell, which is associated with thresher sharks. And that's why I decided to focus on dolphin-associated sets, because my methodology is more impactful for these sets. And I think this is because for dolphin-associated sets, the fishing effort is also a bit more predictable because they go where the dolphins are, and they are usually down here in these areas. Whereas for unassociated sets, they're just trying to find these free-schooling tunas. So the fishing effort is also more variable. And for them to find recurrently inefficient cells throughout the study period was a bit harder. So I think maybe this approach is not the most suitable one for this set, and more important for the dolphin-associated sets.
Okay, and you didn't really talk about regulations in Baja, or like I don't know at all how it is over there. Are there regulations how many times they can go out at sea with the vessels, or like how is it? How can you try to use your data to kind of help regulations?
I was trying to read all these regulations and laws. So the way it works, there's an international framework that I told you about, the IATTC. So they kind of regulate all the tuna fisheries, and they create these measures, and then the national law has to put this into laws. And here we have two authorities, the CONAPESCA, which is the Fisheries and Aquaculture Authority, and the CONAMP, which is establishing marine protected areas. And there are different regulations such as permits and the way they have to handle bycatch. And also most interesting for me was that there's different fishing ban seasons. So actually, they can choose, every large vessel can choose between 2 fishing ban seasons every year. One is during summer, from July to October, and then the other one is during winter from November to January. They made this to give the fishers a bit more flexibility to choose one between one of the two, but then also that means that there's never really a full, well, no pressure on the fishing stocks at all, because there's always some activity. But most of them actually, that's what I show here, most of them chose closure period B, so during the winter months and not during the summer months, which was interesting again, sorry, for my results, because as you saw that most of the inefficiency happened during the summer months and not during the winter months. I think already, again, if we want to simplify the management approach, maybe we could already think about having only one fishing ban season that is most adapted to the ecology. Also considering some social and economic impacts, but then instead of having the two, that could have an impact already and make it a bit more easy than having this adaptive management approach.
And you said, so where you found there was a lot of inefficiency kind of corresponding to breeding grounds and nursery grounds. So that was known already by scientific and like regulations measures. Like they know there is a breeding ground.
They know there's a breeding ground. And so in Mexico, shark fishing is legal as well, targeted shark fishing. And they have also a shark fishing ban, which is from May to August, if I'm not wrong. And so they overlap the shark fishing ban with the reproduction cycle of most of the sharks that are present. But then, of course, if they're bycatch, they're not really part of this regulation. Yeah.
Okay. Thank you.
Maybe we can move forward, Juliette. Sorry to this.
Thank you very much. It was a good presentation, nice strong objectives, and I'm not a specialist, so my questions might be a little bit out there. The grey cells in your figures, and your figures are very nice. What do they really represent? Because are they just places where there's no overlap in the catch, or are they places where fishing activity is lower?
So there is fishing activity if it's gray, and if they are white, there is no fishing activity. I call them efficient here because they're not inefficient. So for simplicity, I called it efficient. However, this simply means that, I mean, to be classified as inefficient, the tuna catch rate had to be below the average, and the shark bycatch rate had to be above the average. But if it's great, that could also mean that we still have a very high bycatch rate, but also a high tuna catch rate, or a low tuna catch rate, but also a low bycatch rate.
The relationship between the two that will determine it. I think it's a problem.
Yeah.
How do you standardize the cells for like percentage of, because at the beginning you talked about how the percentage of bycatch from each species, so how is that standardized within this? Because you have a much higher percentage of bycatch of one shark compared to others.
So you said within the different shark species?
Yeah.
I mean, by calculating the bycatch per unit effort, it should already be normalized.
Standardized. But so on here is, for example, a red cell for a fresher shark, which has a much lower percentage.
Yeah, okay.
Doesn't have the same weight as,
So the average that I compared it to is species specific.
Okay, so you don't care about cross, but these vessels are catching different species, so it does have a little bit of relevance, but I understand why you did that in your analysis.
I mean, I could, show you this map that shows the bycatch per unit effort for the different species, and you can see that for silky sharks we have, for example, a very high outlier up there, whereas for all the other species the values are between 0 and 1.
Representative. Perfect. About the implications for like loss to the industry if they went ahead with your recommendations, like, so nearly 10% of tuna production, how much money are we talking about?
I mean, the thing is that this is what I calculated, but then I don't say that there's a general fishing ban during that period. So the fishers would go just to other areas. So this is not the actual loss that they would have, because they would be fishing in other areas that would be more efficient. So ideally, they wouldn't have any loss at all. Yeah, and there was another study that did the recalculation, the redistribution of the fishing effort, and they showed actually that it wouldn't impact the industry in terms of loss, but I couldn't calculate it to economic values in the end.
And maybe just one last question. You just talked about the vessel overview. What's the impact of this on efficiency? So like mostly they choose one ban. So how does that influence your analysis? Because you did this over 12 months for 15 years or whatever it was. Yeah. So
the fishing ban season. Yeah, I mean, if we look at when this inefficiency is happening for the different shark species, you can see that pressure sharks, they're mostly in March and April, and then silky sharks more during the summer. And as I said, most of them choose the fishing ban from November to January. So obviously,
so this answers why there's such a high percentage of silky sharks.
Yeah. And then also it overlaps with the exact months of the reproduction times of these different species. So thresher sharks, they're more temperate sharks. They are reproducing more during the colder months, and then silky sharks, they are reproducing during these warm months. So yeah, it definitely has an impact. And then I also looked at the CPUE for the different months to see if there's actually a very low CPUE, and that's why they choose these months. But no, it's stable over the months. Sometimes it's even higher in December. Okay. So yeah, I think it would be interesting to talk to them at least as well and see why do they choose this fishing ban, and could they also imagine choosing the other one. Yeah.
Thank you very much.
A couple of minutes for your questions.
Yes.
What happens? How is the bycatch handled? Because I don't understand why in the first place they go to areas that are inefficient for the target species. So if they can sell the sharks, maybe they don't mind having this bycatch. And the second quick question: which other gear types would you include in future studies that have a high impact, high bycatch rate?
Okay, so why do they go there? First of all, as I said, because the dolphins are there as well, and also because this is just a snapshot, but you can see these are a lot of fishing routes. But here is Mazatlan. That's a very important fishing port and fishing industry area. So most of the vessels come from here. So it makes sense for them to stay kind of close to their port and fish there. And then if they have bycatch, the IATTC mandates to release them as quickly as possible if they're still alive. But then sometimes, obviously, they are already dead by the point that they're on the vessel, or maybe they don't see them. So it can still happen that they have them on the boat. Now it's not allowed to do shark finning anymore. So they have to bring back the whole shark to the port. And then I was talking to a shark scientist in the area, and he said that often, I mean, obviously you could bring it back and sell it as well, but bringing back the whole shark is a huge animal, and it takes up a lot of space and weight. So usually for them, it's more economically, it makes more sense to bring back more tuna and put the shark back at sea, even if it's dead. Because, yeah, so and then there's different species-specific regulations for the different bycatch species, but maybe I'm not going to go into details for all of them.
And the other gear types?
What other? I would like to look at longliners, because it's commonly known that longliners actually have a higher bycatch rate than purse seiners. The problem is that the data for longliners is only available at a 5-degree resolution, so that would be too big. So I think also actually my methodology could be an example of how we could analyze longliners in case we get the data at one point in the future.
No, amazing. I love the topic and really well done.
Thank you.
Maybe a last question.
Yeah. Well, everyone's.
Can you know more about how you then give underreporting data? It's pretty smart that this IATTC puts observers on the vessels, but I also know sometimes these are not very well paid, maybe, so there's corruption cases. I wanted to know how these observers are selected, and in general, underreporting data really are not a problem in the sample.
So what about the observers and the potentially unreported catches?
Big topic. I was never on the boat, so I cannot tell. I know I'm aware of the issues, and I can imagine that there is probably some unreported data that's done under a lot of pressure as well. But I really can't judge because it's not my own data, so I couldn't include it in my analysis. What I can say is that this is what is happening, at least. Like, this is kind of a precautionary approach because that's what is reported. It might be more as well. Since in the end, I'm not really focusing on the specific numbers, like the absolute numbers of bycatch, but more looking at the areas. I think, of course, it matters and it would be interesting to know, but the scope of this study was also to identify these hotspot areas. So that's not that much dependent on the number.
There were no corruption cases in the news or things like that. Just like not a TV, let's say, things like that.
On the news, I'm not sure. I haven't read anything. Talking to people, potentially.
Okay, Alex, your last question.
You showed a graph with the different catch rates of tuna itself, like all the different species. Is there a difference between catch rates for different tuna species? And does the tuna abundance of each species differ to the targeted catch? It's like, I'm sorry, it's like 2 questions.
No, yeah, don't worry. So we have the different shark bycatch proportions for the different set types, and then the same graph for tunas. So we see that for dolphin-associated sets, it's mostly yellowfin tuna, like 97%. And so we know that yellowfin tuna are usually associated with dolphins. And it is interesting because for them, economically, it makes a lot of sense, because for a yellowfin tuna to keep up with the speed of a dolphin, they have to be pretty big. So they're also catching large individuals in that set type. And then for unassociated sets, since they're targeting free-schooling tunas, everything is more mixed. So the tuna catch, the species composition is more mixed, and then also the shark bycatch proportions are more mixed. Mm-hmm.
Do you know by any chance what the abundance of the tuna species is like? I think, if I remember correctly, I think skipjack usually they're much more abundant than yellowfin, if I remember correctly, for example. But was there anything that you looked at in that direction?
I mean, what I looked at was let me find this here. It was the CPUE for the different species in the different regions. So you can see a spatial segregation. I mean, as I said, for dolphin-associated sets, it's yellowfin tuna anyways. Then for the unassociated sets, it is mixed, but it is also spatially segregated. So we have more yellowfin tuna in the south, more skipjack tuna in the Pacific Ocean, and then in the Gulf, it's mostly black skipjacks. So, yeah.
Thank you for your answers. Sorry. And you can, of course, continue the discussion. That's what's nice. Thank you and congratulations, Lena.
Thank you.
And you can take them out to... Yeah. We're going to finish with some props and see. Yeah. Apparently, Jakob needs to set up the scene. It's generally at that time that I take the opportunity to thank the supervisors, but I will find myself in a weird position. So I would like to thank Hubert Bonnefond for co-supervising. But we were actually mentors as part of a feasibility study and not supervisor. And of course, I'm not assessing anybody this week.
Naturally.
Are you ready?
Yes, I'm ready.
The camera doesn't seem to
be
ready.
Yes.
Yes.
Very good. Okay.
All good when you're ready.
Okay. Do I have the right? Yes, I do. Hello and welcome to this very last presentation of the 2026 MARRES Symposium. My name is Jakob Roeckl, and I'd like to present to you today my feasibility study on fish leather gloves, or in more detail, the question of assessing the feasibility of producing leather gloves from tanned fish skin, which is a special material. We'll talk about it in a moment. And to set this in the context of MARRES, it's about. At this point, I'd like to give a huge thank you also to the expert at Roeckl and for my supervision from Christophe and Hubert. And thank you for listening.
Let's open the floor to questions to Aziz first, following Robin. So followed by Robin.
I think we could successfully sell this product.
Thank you, Jakob. Let's circle back on some other aspects we've seen also yesterday. Thank you. To conclude this MARRES Symposium, congratulations, Jakob.
I'll just put the slide on.
And somehow we managed to finish with only 5 minutes. 5 minutes late. So I would like also, of course, to thank all the jury members and reviewers and the supervisors for all your work. For you, ladies and gentlemen, for having been there for the whole period, including the blue manager who finished yesterday, but who are here, at least you. And so it has been a great pleasure as director of the program to come back. For you to come back and to see all of your work. I said that at the beginning, early, really, like you were not students anymore. You were having science to share between each of you, and seeing you also talking about that as part of other things during the breaks. It's fantastic. Of course, big thank you to the team who have been there, Christelle and Diana. Thanks a lot. And I'm sure he will be delighted. I will give the floor to Aziz, who has been there the whole time. So we asked Aziz, yeah, where are your notes? Where is your speech? So Aziz had also a special connection with you as he entered this education with MARRES when you arrived. From the breakfast club, he has kind of started, and it was a big legacy from this year, to a lot of classes from ecology and others. So yeah.
And R.
Yeah, I was trying to hide the R stuff.
I'm super impressed by the level of presentation. I think I said to Andre yesterday, it's super difficult to grade because we were quite lucky when you came in that you were already good, which shocked us. So that's why we really have to kind of go tough on the grading. But despite that, the level that you having like increased in your ability is impressive, is really impressive. And I know you all come from different backgrounds and you have different passions and things like that. And it's really difficult to be the constant here and not try and be influenced by what I enjoy and what I don't enjoy. And I promise you, I wasn't and stuff. But I just want to say I'm very proud. Like Christophe said, like I came into the program the same time as you, and you may not have noticed, but I was super nervous to teach you guys as well, especially 19 girls and 2 poor guys there as well. But you really allowed me to do my job and become a better teacher as well, and that's hopefully going to benefit me. So congratulations to you all, and thank you.
Isn't it a weird setting to be like that?
It's been weird all week.
Yeah, that's true. Okay, so sorry for the jury members. We're going to come back to you. Also, thank you for all of you who are following online. I will now end the stream and talk about serious stuff, about what's going next, and this is private. So yeah, for what's going next, so we will nicely ask, kindly, firmly ask you to leave the room and have your life for a few hours. Just hold on. This afternoon, Christelle, at what time do we meet in this bar and where?
I'm sure they're heading straight.
Sorry, we are still streaming.